This is Professor R.M. Mahendrakar from JSS Arts, Science and Commerce College, Gokak. We all know that proteins are one of the most important biomolecules present in all the living organisms. Proteins are the major biomolecules which are responsible for the functioning of the organism and also for the structure of the organism. So how these proteins are synthesized in the cell, that is what we are going to study in this video. So for convenience, I have divided this topic into 4 parts. In part 1, we'll be dealing with the process of transcription in prokaryotes. So let's start with this topic on protein synthesis. DNA present in the nucleus directs protein synthesis, but actual synthesis of protein occurs in the cytoplasm with the assistance of transfer RNA and ribosomes. The whole process of protein synthesis takes place with the help of 2 main steps, that is transcription and translation. During transcription, the genetic information present in the DNA is transcribed into mRNA molecule. The mRNA molecule is then translated in the ribosomes into protein molecules. This unidirectional flow of information from DNA to mRNA and then to proteins is called the central dogma in molecular biology. Now, in organisms where the genetic material is RNA, like the retroviruses, there the RNA first synthesizes DNA, that is the genetic information on RNA is transferred to DNA through a process called as reverse transcription, and this was discovered by Thiemann. Hence this phenomenon is known as Thiemann. So this whole process we call it as central dogma reverse. So let me explain. Now this is a DNA molecule. Now during protein synthesis, the whole DNA molecule is not copied. Only the part of the gene, that is the genomic part of the DNA, the gene that is required. So that information is transcribed on mRNA through this process called as transcription. Then this mRNA, which is carrying the genetic information in form of these codons. Now these codons are translated and then it results in formation of a protein molecule. So this process we call it as translation. So you see the genetic information from the DNA, it is moving to mRNA, and from mRNA it is moving into the protein molecules, and this protein molecule may behave as a functional protein molecule or a structural protein molecule. Now, in case of organisms where the genetic material is RNA, like for example this coronavirus, there the genetic material is RNA, it is multiplying in the bodies of human beings. So there we see this Thiemannism or central dogma reverse process. Means what happens here is, whatever the genetic information that is present in this RNA is first transcribed to DNA molecule by a process called as reverse transcription, and then this DNA molecule then transfers that genetic information into the mRNA through a process called as transcription, and then this mRNA then synthesizes the required protein through a process called as translation. So this whole process, we call it as central dogma reverse, this protein synthesis in detail. So before that, first let's begin with this first step, that is transcription. So a transcription unit is primarily composed of three regions in the DNA. All the transcription units will have these three regions. The first is a promoter. It is located at the 5' end of the structural gene. Second is a structural gene, that is the gene which is going to be transcribed on mRNA to synthesize the protein. And then we have a terminator, that is to stop the synthesis of mRNA, which will be located at the 3' end. Now here you see this is a DNA molecule. Now DNA is a double-stranded molecule. So in this double-stranded molecule, both the strands cannot act as a template for transcription because DNA-dependent RNA polymerase catalyzes the polymerization in only one direction, that is 5' to 3' direction. Hence, the DNA strand that has the polarity 3' to 5' acts as a template and is also referred to as the template strand. So this strand on which the mRNA will be synthesized, we call this strand as a template strand, and the opposite strand which is not taking part in transcription, we call it as a non-template strand. Now as I've told you, there are three regions on this in this transcription unit. Now this is the promoter region. The promoter region is towards the 5' end of the structural gene, and this is the structural gene. So this is the gene that will be coded, so that's why we call it as RNA coding region. And then this is the transcription termination region. So here the transcription will be terminated. Now the part of the DNA molecule towards the left side from the first nucleotide to be synthesized, we call this end as upstream end. And from the first nucleotide that will be synthesized on mRNA till the termination, this end we call it as downstream end. Please remember this because we'll be using these terms. Then this RNA coding region, then through a process called as transcription, it synthesizes this mRNA. So this mRNA will be having the coding regions and a terminating region. Now here in protein synthesis, we refer only to mRNA. But remember, all the different types of RNAs, all are synthesized through this process called as transcription. So let it be ribosomal RNA, let it be transfer RNA, let it be messenger RNA. All these are synthesized through a process called as transcription. Now let us see the transcription in prokaryotes. The process of synthesis of RNA by copying the template strand on DNA is called transcription. As I've told you earlier, cells contain several types of RNAs, mainly ribosomal RNA, transfer RNA, messenger RNA, and in eukaryotes, we have some additional RNAs like small nuclear RNAs, and these are actually responsible in mRNA splicing. This I'll tell you when we deal with the transcription in eukaryotes. But in prokaryotes, we don't have this. We have these mRNAs, tRNAs, and rRNAs. So all these are transcribed on the DNA segment through a process called as transcription. But remember, in transcription, only the selected portion of the genome is copied. The enzyme involved in transcription is RNA polymerase enzyme. Unlike the DNA polymerase, it can initiate transcription by itself. That is, it doesn't need any primase here. And more exactly, it is a DNA dependent RNA polymerase enzyme. Now, the steps in the transcription of prokaryotes, we segregate them into three major steps. First is the initiation. Now, initiation involves these three most important events. One is a closed complex formation, open complex formation, and third is the tertiary complex formation. Then we have this elongation step, and then we have this termination step. Now, in prokaryotes, there are two types of termination. One type of termination is row dependent, and the another type of termination is row independent. So now we shall see each of these steps in detail. Bacterial RNA polymerase is the principal enzyme. There is a single RNA polymerase enzyme in bacteria which transcribes all the different types of RNAs. This single RNA polymerase enzyme is called the core polymerase, and it consists of 2 alpha units, 1 beta unit, 1 beta prime unit, and 1 omega subunit. The binding of the core polymerase to promoter is facilitated by a specified subunit called as the sigma factor. So when sigma factor binds with the core enzyme, it forms a holoenzyme. So thus, in case of this E. coli bacteria, the sigma factor 70 facilitates the binding of the RNA polymerase to the promoter region. Now we shall see the function of each of these subunits of the holoenzyme of RNA polymerase in E. coli. The alpha subunit determines the DNA to be transcribed. The beta subunit catalyzes the polymerization and helps in synthesis of the mRNA. Beta prime binds with the DNA and opens the DNA template. The sigma factor recognizes the promoter for synthesis of the mRNA, and the omega unit helps in the packing of the subunits. The binding of RNA polymerase to promoter is facilitated by the specified sigma factor. For example, in E. coli, it is pi 70. Sorry, sigma 70. So the sigma 70 factor identifies the promoter site in E. coli. In E. coli bacteria, the promoter site consists of two conserved sequences. At the 5' end, here we have this TTGACG region, which is minus 35 base pairs away from the starting region. So this is in the upstream side. Then this region, that is TTGACG, we call this region as the recognition sequence. And at the -10 region, there is another sequence. It is TA-TA-AT region. Now, this region we call it as Primnow box. Now, these sequences are upstream to the site from which the transcription begins. Now, binding of the holoenzyme to these two conserved sequence of the promoter forms a complex. Now, you can see here. Now, this is the RNA polymerase enzyme. Now, this is the sigma factor. Now, this is the Primrose box, which is at the -10 upstream region. This is the recognition sequence, which is at -35 base pair sequence in the upstream side. And so these are the two binding, and then this we call it as the closed complex. Then after formation of the closed complex, the RNA polymerase holoenzyme separates 10 to 14 bases extending from -11 to +3, and this process we call it as melting. Now, so from here, from -11 to +3, now these nucleotides are separated. So this region we call it as melting, and this now forms the open promoter complex, and the initiating nucleotide gets exposed. So this whole process we call it as isomerization. After the isomerization step, the RNA polymerase starts synthesizing RNA nucleotide strand based upon complementary pairing. If the enzyme synthesizes short RNA molecules of less than 10 base pairs, it does not further elongate. This is called abortive initiation. This is because the sigma acting as a mimic of RNA, and it lies at the middle of RNA exit channel, and hence the initiation process stops. But when RNA polymerase manages to synthesize RNA nucleotide strand of more than 10 base pairs, it ejects the sigma region from the RNA polymerase, and the RNA further elongates and exit from the RNA exit channel. So this results in formation of this tertiary complex. So thus the initiation process ends here. So in initiation, so after the sigma factor is ejected, the enzyme moves along 5' to 3' direction continuously, synthesizing mRNA. The synthesized mRNA exits from the RNA exit channel, so as you can see here. Now, during elongation, the RNA polymerase moves along the DNA molecule, melting and unwinding the DNA helix as it progresses. The enzyme adds ribonucleotides to the 3' end of the growing RNA molecule, with the order of addition determined by the order of the bases of the template. strand. Each elongation takes place by addition of activated ribonucleotide triphosphates, that is ATP, UTP, GTP, and CTP, to one strand of the DNA template. For each nucleotide added to the growing RNA chain, pyrophosphate is given out. This is rapidly hydrolyzed to inorganic phosphate. The synthesis of RNA chain is energized by the expenditure of energy in form of pyrophosphate. For each nucleotide monomer added to the chain, two high energy phosphate are expended. Elongation of the RNA chain takes place by the means of the core enzyme, which moves along the DNA template during transcription. RNA is synthesized by the polymerization of the ribonucleotide triphosphates. The third prime OH of one ribonucleotide reacts with the 5 prime phosphate of another to form a phosphodiester bond. The transcript is synthesized in 5 prime to 3 prime direction. But because the chain must be anti-parallel for the base pairing, only the 3 prime to 5 prime template of the DNA can take part in the transcription. So this process of elongation continues till the ribRNA polymerase enzyme comes in the vicinity of the terminating region. So once it comes into the terminating region, then the third step, that is termination, begins. As I've told you, in prokaryotes, there are two mechanisms of termination. One is row independent and one other is row dependent. First, let us see the row independent type of termination. In the row independent termination, the transcription is terminated due to specific sequences in the terminator DNA. The terminator DNA contains invert repeats, which causes complementary pairing. Now here you can see these are the invert repeats. Now here we have this GGTCGACTG and its inverse repeat is here GGTCGACTG. Similarly like this. So all the terminating ends have these invert repeats. So when a mRNA is synthesized on using this template, now this is the 3 prime to 5 prime end. So here we have the synthesis of this mRNA based on the complementary pairing. So once this RNA is synthesized, then what happens is this G will form a complementary pair with this C. This G will form a complementary pairing with this. So because of this complementary pairing, what happens here is a hairpin structure is created. And because of this hairpin structure, the load of this hairpin structure is not tolerated by the AU base pairs. So RNA gets separated from the RNA DNA heteroduplex, and thus the RNA molecule is released from the RNA polymerase enzyme. Now in case of this row dependent, now in row dependent type of transcription, there is this row factor. The Rho factor, it is a hexamer ring-shaped single-stranded binding ATPase protein. So, as this mRNA is synthesized, the Rho factor attaches to the recognition site of the mRNA and moves along it. Then, when the RNA polymerase enzyme comes in the vicinity of this terminating region, it pauses. And then the Rho factor hydrolyzes the mRNA from the RNA polymerase enzyme, and all these three are released from the DNA strand. So thus the RNA and the polymerase enzyme and the Rho factor are released after termination. You should remember that the Rho protein does not bind to those RNAs whose protein is being translated. Rather, it binds to the RNAs after translation, because in bacteria, the transcription and translation, these processes occur simultaneously, because they are all taking place in the cytoplasm. So when translation process is going on, the Rho factor doesn't bind with the RNA. Only once the translation process ends and still the transcription process is going on in case of this prokaryote, then only the Rho factor will bind with the mRNA. Now let us summarize the whole sequence of transcription in prokaryotes. Now this is the RNA polymerase enzyme, this is the core enzyme. Now when the core enzyme binds with the sigma factor, it becomes a holoenzyme. The holoenzyme binds with the DNA whose segment has to be transcribed, and the sigma factor identifies the promoter region, and then this forms a closed structure. Then some nucleotides are cleaved, then becomes an open structure, and once the mRNA is synthesized, the sigma factor is released. This becomes a tertiary structure, and after that the elongation process begins, and a new mRNA strand is synthesized based upon the complementary pairing from the template strand, because in DNA only the 3' to 5' DNA strand acts as a template strand. On this strand, the mRNA is synthesized from 5' to 3' direction, and the nucleotides are added to this mRNA based on complementary pairing. And then once it comes in contact with the terminating region, then the RNA synthesis stops, and then the RNA is released, and then this RNA then undergoes the next process, that is translation. So thank you. In the next part, we shall see about the transcription in eukaryotes.